Technical Veterinary Learning Channel, honored by Professor Ahmed Mamdouh Sharif, Professor of Epidemiology and Infectious Diseases, the ex-head of Department of Veterinary Medicine, the ex-Vice Dean of Faculty of Veterinary Medicine, Bani Suef University, Egypt, presents today series of epidemiology to be completed. Fifth episodes of Principles of Prevention and Control of Infectious Diseases will discuss completely with examples a very important subject about vaccination program and vaccines. Vaccination program, it means a plan to apply vaccination to an epidemiologically appropriate proportion of susceptible animals or human population for the purpose of disease prevention and or control. Objectives and strategies of a vaccination program should be defined by the veterinarian and or health authorities before implementation of vaccination, taking into consideration epidemiology of the disease, its impact and zoonotic potential, the species, sexes affected and their distribution. There are two types of vaccination program: systemic or mass vaccination and emergency vaccination. Mass vaccination often used in attempt to reduce the prevalence of endemic diseases to the levels that allows selective slaughter and or depopulation can be used in prevention and control of any disease. Mass vaccination facing some difficulties that may arise with the possibility of screening tests that used to distinguish between natural infection and vaccine induction of immunological responses, such as foot and mouth disease vaccine, RB51 vaccine, BCG vaccine, and etc. Emergency vaccination. It means a vaccination program applied in immediate response to an outbreak or increased risk of introduction or emergence of a disease. This is an emergency preparedness, and this, like what's used in the world now for prevention and control of COVID-19 or SARS-CoV-2, that all the vaccines as AstraZeneca, Pfizer, Sinovac, or the Russian vaccine are licensed in WHO, Gavi organization as emergency vaccination. It provides an adjunct to application of other essential biosecurity and disease control measures and may be applied to control outbreaks. Emergency vaccination may be used in response to the following. Number one, an outbreak in free country or zone, like what's going now at 2021, from introduction of emergency vaccines all over the world countries to vaccinate the people against COVID-19. Number two, an outbreak in a country or zone that applies systemic vaccination, but when revaccination is applied to boost existing immunity, such as the revaccination every 4 months that done in countries infected by foot and mouth disease for their local strains. Number three, an outbreak in a country or zone that applies systemic vaccination, but when the vaccine employed does not provide protection against the strain of the pathogenic agent involved in this outbreak. The most clear example here is the new vaccine of foot and mouth disease that contains the newest strain of foot and mouth disease that present in Egypt during this time. Number 4, a change in the risk of introduction of pathogenic agent or emergence of a disease in a free country or zone, such as Zika disease that introduced in the south of United States coming from middle and south of America, which change the risk of introduction, so have to put emergency vaccination to counterattack of that infection. Systemic or routine vaccination. It's aiming in endemic to reduce incidence, prevalence, or impact of a disease with objective of prevention, control, and possible eradication of any disease. Systemic or routine vaccination. Its objective in free countries or zones is to prevent introduction of a disease from an infected neighboring country or zone, or to limit the disease impact in the case of introduction of that disease. The most clear example here for this is the vaccines against African horse sickness that used in the south part of Egypt to protect this country from introduction of this disease from the neighboring Sudan state. Vaccination strategies. Different vaccination strategies may be applied alone or in a combination, taking into the account epidemiological and geographical characteristics of occurrence of a disease. There are 4 vaccination strategies. Number 1 is the barrier vaccination or frontier vaccination. Means vaccination in an area along the border of an infected country or zone to prevent the spread of the infection into or from a neighboring country or zone. Both examples are Zika vaccine that used in the south of United States, as well as African horse sickness vaccine that used in the south of Egypt. The second vaccination strategy is the blanket vaccination. Means vaccination of all susceptible animals and or persons in an area or an entire country or zone. The most clear example in Egypt is the vaccination of foot and mouth disease vaccine to all ruminants, and for health status of human, vaccination of all children under 6 years with polio vaccine. Ring vaccination is the third strategy of vaccination, which means vaccination of all susceptible animals in a defined area surrounding the location where an occur of outbreak was noted, such as ring vaccination with PPR for the sheep at an area of 25 kilometers around infected area in Egypt. The fourth type of vaccination strategy is the targeted vaccination, which means vaccination of a subpopulation of susceptible animals, such as use of Nemo 5 or Nemo 4 to vaccinate the calves in the early stages of their lives to protect them against 5 and or 4 diseases that threaten their life. Exit strategy of vaccination program. The vaccination program may provide for an exit strategy to ease vaccination. The cessation of vaccination may apply to the entire target population or to a subset of it. The most clear example is the using of vaccines against brucellosis. Exit strategy of a vaccination program criteria to ease vaccination may include the following. Number 1, eradication of the disease in a country or zone has been achieved, such as when we want to eradicate the brucellosis from Egypt or any country with high risk of it. Number 2, risk analysis demonstrates sufficient reduction of likelihood of introduction of a pathogenic agent or emergence of a disease, such as lumpy skin disease when it enters to a free country. Number 3, reduction of the incidence, prevalence, or impact of a disease to level where alternative measures such as stamping out policy may be more appropriate to achieve disease, such as foot and mouth disease eradication in United Kingdom at the beginning of this millennium. Types of vaccines. Number 1 are the living vaccines. There are 4 types of living vaccines. Number 1, attenuated modified vaccines. Vaccines are prepared by different methods like the following. Number 1, adaptation of the organism to unfavorable conditions such as growing of Bacillus anthracis in 50% serum agar in atmosphere rich in carbon dioxide. Number 2, passage in unusual hosts such as passage of rabies virus in a chicken embryo. Number 3, passage in tissue culture such as passage of rinderpest virus lymphotropic in bovine kidney cells for 90 to 120 passages. Number 2 of living vaccines are naturally virulent vaccines, Brucella abortus strain 19. Number 3, virulent organisms such as contagious pustular dermatitis vaccine. Number 4, antigenically related organisms, heterotropic vaccination such as the use of sheeppox virus vaccine to protect cattle against lumpy skin disease. Number 2, inactivated vaccines. These vaccines are made from virulent organisms by destroying their infectivity using inactivating agents or physical means and retaining their immunogenicity. Inactivated vaccines used to induce immune response of lesser efficacy in comparison to living vaccines. In addition, they need to be administered in large doses and multiple injections. They also require the incorporation of adjuvant to increase their antigenicity and prolonged sensitization of the host. Genetically engineered vaccines. In these vaccines, certain genes are deleted. Deletion of the gene of virulence, as in Egyptian disease virus vaccine, as well as in AstraZeneca vaccine against COVID-19. Recombinant vectored vaccines. In these vaccines, the genes encoding for important protein antigens of an organism are cloned into the genome of another vector organism, such as pox virus or herpes virus. The resulting recombinant organism can express such genes and to produce the desired antigens upon vaccination of the host, such as vaccinia rabies G glycoprotein recombinant vaccine. subunit vaccines. These vaccines contain immunogenic component of an infectious organism. The gene coding of antigen of interest is isolated from the genome of the organism. This gene DNA sequence is inserted into an expression vector such as E. coli bacteria or yeast. The recombinant antigen is expressed in vitro through the protein expression of host vector, such as feline leukemia virus recombinant subunit vaccine by using of E. coli vector. Vaccination failure. Several factors have been reported to be responsible for failure of vaccines to confer protective immunity as follows. Number 1. Using of inappropriate serotype of vaccinal pathogen in vaccination against diseases caused by antigenically distinct serotypes, such as foot and mouth. Number 2. Exposure of thermolabile living viral vaccines to high ambient temperature during storage or transportation, such as attenuated tissue culture vaccine of rinderpest and or lambskin disease. Number 3. The use of inefficient vaccine due to reduction in immunogenicity during inactivation. Number 4. The use of under-attenuated vaccine that may result in induction of the disease. Number 5. Development of non-immunizing mutants of the vaccinal virus during vaccine preparation. Number 6. The use of improper dose of vaccine and unsatisfactory administration. Number 7. Inactivation of living vaccines by excessive alcohol swabbing of the skin. Number 8. Vaccination of animals incubating the disease itself. Number 9. Concurrent infection of vaccinated animal with immunosuppressive viral infections such as bovine viral diarrhea and AIDS. Number 10. The conjunction use of antibiotics with living vaccines that leads to inactivation of the vaccinal organism. Number 11. Vaccination of animals under immunosuppression due to mycotoxin or some viral antiparasitic affections. Number 12. Presence of stress factors such as malnutrition, internal parasitic conditions, pregnancy, and extremes of cold and heat. Number 13. Presence of passively derived maternal immunity in young animals through colostrum. Number 14. And vaccination of very young animals before the full development of immune system. Certain diagnostic screening tests can be used for spotting out carrier animals in the herd. These tests should be periodically conducted on an all animal in the herd so that carriers can be diagnosed and culled. Series of principles of prevention and control of infectious diseases to be continued.